You see it. Great. Excellent. Okay, so thank you, team, for the introduction. And I thank the organizers for inviting me to share our experience with the SyncroPatch, using it for our research, which we have been doing for more than a year now. And first, as some of you know, light-gated ion channels are used for optogenetic control of cellular excitability. Their genes are fused with cell-specific promoters, encapsulated into virus particles, and injected in the brain. And once they are expressed, illumination of the brain will cause either activation of the target neurons or inhibition, depending on the type of light-gated ion channel that you use. And this would cause changes in the animal's behavior. But of course, what is most important, that there are several ongoing clinical trials of using light-gated ion channels to restore vision in blind patients, and there was one paper already published on it. And light-gated ion channels are also considered as gene therapy for many neurological, psychiatric, and cardiac disorders. So they are really important. Synthetic light-gated ion channels are made by fusing light-sensitive domains of other proteins to endogenous neuronal channels, but by far the most popular optogenetic tools are light-gated ion channels that naturally occur in protists. And these proteins are called Channelrhodopsins because they are retinal proteins, just like visual pigments in our eyes. But they, upon photoexcitation, they generate transmembrane ion currents, passive transmembrane currents, rather than activate enzymatic cascades. And Channelrhodopsins are the only so far natural or any proteins known to do so. In our lab, we do not run actual optogenetic studies in animals. Our job is to provide molecular tools for that. And different optogenetic studies require different tools, different in the in ion selectivity, in photocurrent kinetics, in action spectra, and so on. And Oleg Senyshchikov in our lab demonstrated that the first discovered Channelrhodopsins, and it was more than 20 years ago, act as photoreceptors in algal phototaxis. And since then, we and others have identified hundreds of Channelrhodopsins in various protists. And only few of these Channelrhodopsins have been characterized electrophysiologically because of low throughput of manual patch clamp. And that was the moment that, that we decided that we need an automated platform for current clamp, for patch clamp recording. And Tim has already introduced the SyncroPatch to you, and we have worked with two models of it. So we got the Nanion instrument grant to work with model 384I, which we used to collect preliminary data to apply for an S10 grant to get the most recent model, which is 384 without the I. And as Tim already mentioned, this model has several improvements over the previous version. And for us, really important is this optional partial chip usage that Tim also has mentioned. We had 6 labs from 4 research institutions in the Houston area to participate in our Nanion instrument grant. And these people work with really different channels in different cell types, and they all have been able to collect enough preliminary data for the S10 grant, and we also invited 6 more labs to this endeavor. But today, I will tell you only about our studies on Channelrhodopsins and in HEK cells. Using the SyncroPatch was my first experience with automated patch clamp, and I was afraid that, okay, automated patch clamp, it means it would take a lot of cell culture, but in fact, one standard 10-centimeter plate is sufficient for 2 SyncroPatch experiments, so it's not so bad. But of course, adherent cells have to be enzymatically dissociated before measurements. As Tim has already explained, the SyncroPatch uses this planar patch clamp format, and very important that the chips are available with different number of cells, different number of holes, so you can either record photocurrents or currents from one individual cell, or the currents are summarized to improve the signal-to-noise ratio. And of course, there are chips with different resistances to be suitable for different cell types. And these multi-hole chips are particularly relevant for us because, of course, we cannot generate stable cell lines for each of hundreds of Channelrhodopsins that we want to test. So we use chemical transfection, and chemical transfection is never 100%, as you know. And here you see some results of comparing a 1-hole chip with a 4-hole chip for the same Channelrhodopsin. And each little square here represents photocurrents from one well. And there is also a programmable quality control, a very important feature of the SyncroPatch. And in this case, green squares mean that this well has a seal resistance above 500 megohms, and which is a good seal. And gray squares means the resistance was lower than this value. And you can see that in both cases of 1-hole and 4-hole chips, the success of gigaohm seal formation was very high, but in the case of 1-hole chip, many green squares actually show no photocurrents, and that's because the captured cell did not express Channelrhodopsin. But in the case of the 4-hole chip, almost all green wells showed great photocurrents, and their amplitude was, of course, also larger. Nanion provides standard solution for the SyncroPatch measurements, and their external solutions are indeed pretty standard, but their internal solutions are quite unusual because they are based on fluoride salts. And these fluoride salts are a key ingredient for the high success rate of Gigaseal formation because of formation of insoluble calcium fluoride. So as you all know, depolarization of the membrane may usually decreases the seal resistance, but it's not the case with the SyncroPatch solutions, as you can see on the left. And on the right, you see that these seals, gigaseals, are also very stable upon repetitive application of the same voltage protocol. And the new model of the SyncroPatch also provides an option for fluoride-free recording, but we have never used it because all our channels are either conduct fluoride or are indifferent to it. Now, this slide shows something that most of you will not need in your experiments, namely the photostimulation unit of the SyncroPatch. It is still under development, and currently it can illuminate only 1/4 of a chip, and that's why we need this partial chip recording mode, because we have to repeat our measurements 4 times to cover the entire chip. It consists of, the basis of it is an array of light-emitting diodes, And you see that Nanion team, Nanion engineers have worked really hard to improve this system. They optimized both the hardware to have more output light intensity and the software to improve synchronization between the triggering of LEDs and data acquisition. In the previous version, you see here that sometimes the light pulse was triggered prematurely, which is, of course, bad for automated analysis of the data. And it makes no sense to have throughput data acquisition if you need to analyze each trace individually, manually. But the latest version provides a perfect synchronization, and this is just an example of really fantastic technical support that Nanion provides. So, Channelrhodopsins, they are not only numerous, but also very, very different. And there are 4 structural and functional classes of them. Anion channelrhodopsins, or ACRs, generate chloride influx and are used to inhibit neurons. There are 2 different families of cation channelrhodopsins, or CCRs. These generate proton and sodium influx and are used to activate neurons. And the most recent addition to this set are calcium channelrhodopsins, or KCRs. They are potassium selective, but we couldn't call them PCRs because that would be very confusing. And they are also used to inhibit neurons. And as we have just received a notice that our paper reporting a structure of KCRs has just been accepted to Nature Neuroscience, so it should appear in the press very soon. Channelrhodopsins are either dimers, shown on the left, or trimers, shown on the right, but each protomer is individually capable of ion conductance, which of course is very different from the classical voltage or ligand-gated channels. To give you a feeling of how channelrhodopsins work, I would like to show you this short movie from the lab of Adam Claridge-Cheng, who was the first to use ACRs in their system after we have published them. And here, ACRs, two different ACRs, or a previously available tool that is a chloride pumping rhodopsin, are expressed in the motor neurons in flies. And you look at a container with flies from the side, and when colored bars will appear on top, this means that the light is switched on. And when the bars disappear, this means that the light is switched off. So let me start the movie now. So in the dark, the flies are just crawling around, and now the light is on, and you see that in both samples with ACRs expressed, the flies paralyze and fall off to the bottom, but they are not dead, because after a while the light will go off, and they will recover and start crawling again. And you see that in the rival tool, with the pump, it's just not efficient. Here, the light is off, and they start crawling again. And besides the flies, ACRs have been used successfully in C. elegans, in the zebrafish, in rats, in mice, in ferrets, and in monkeys. And there are maybe at least 70 published studies of using them as optogenetic tools, but there is a catch. As I said, that ACRs generate passive chloride currents, which means that their direction depends on the electrochemical gradient of chloride. And in the soma of mature neurons, the chloride concentration is low, so ACRs generate chloride influx, and you can use them for inhibition. But in some axonal terminals, the chloride concentration is high. And in this case, ACRs can depolarize the membrane and cause neurotransmitter release, which is, of course, bad. So the current voltage relationship of the wild-type ACRs is pretty linear. And so this means that one way to overcome this problem of axonal stimulation is to generate an outwardly rectifying version of ACRs. And we tried to do this. It's not easy, but we could find some mutants that show outward rectification using manual patch clamp, but those mutants generated very small photocurrents at any voltage. And of course, this cannot be used as optogenetic tools. So we teamed up with a group of Francois St-Pierre, and they developed in Baylor College of Medicine, and they developed a high-throughput fluorescent screen for rectification, and they claimed that they could select outwardly rectifying ACRs mutants with this assay. But of course, these mutants need to be verified by patch clamp, and here is when we used the SyncroPatch. We tested many mutants, and here you see the results of some of them. And of course, because we knew from our previous studies that ACRs, the permeability of ACRs to fluoride is less than to chloride. And that's why the reversal potential of ACR photocurrents with SyncroPatch solutions is negative, about minus 40 millivolts, but nevertheless, we could easily detect rectification when it is present. And here there are examples of some new mutants that we verified by the SyncroPatch. And what is good, that some of these new mutants are really better than the ones that we selected by manual patch clamp, and they are also included here. And here on the right, the upper panel shows you the rectification index, which is basically the ratio of chloride influx to chloride efflux. And the lower panel shows you normalized photocurrent. And you can see that at least two mutants showed here in red are better than our previously selected mutants that really generate tiny currents. And right now, our other collaborators from Baylor College, neuroscientists, they test these new mutants by expression and recording from brain slices of mouse. I mentioned that cation channelrhodopsins conduct not only sodium, but also proton. And this is, of course, bad because in this case, illumination can lead to acidification of the cytoplasm in neurons. And this is something unphysiological we want to avoid. So we use the SyncroPatch to screen new CCRs for reduced proton to sodium permeability. And on the top, you see control experiments with Channelrhodopsin 2, that is the most popular neuronal activation tool currently used. And you can see that changing of the external pH causes very large shifts of the reversal potential, which means that there is a strong permeability to protons and the cytoplasm will be acidified. The lower panels show you the results recorded from some new CCR. And you see that this protein, this channel, shows a much smaller shift of the reversal potential, which means that it should not create any or very little cytoplasm acidification. So this also needs to be verified by experiments in neurons. And finally, KCRs, potassium selective channelrhodopsins, they lack the potassium selectivity filter that is commonly present in all classical potassium channels like voltage or ligand-gated channels. So the question is, what are structural determinants of their alternative potassium selectivity? And we want to identify the responsible residues, and this, of course, means making and testing many mutants. And again, SyncroPatch is very useful for that. And here on the left, you see photocurrents recorded from the wild type and from one of the mutants that shows a really great change in the reversal potential, which means that it's basically sodium selective. And on the right, there are the results from many other mutants. And you can see that the SyncroPatch can really be very informative in this respect. This is our ongoing research because there are still many more mutants to test, but we hope that finally we get to the core of it, and we will know why potassium is selected by this type of channels. This, I think, was my last research slide, and I would like to thank our research team and our collaborators. And last but not least, I would like to thank the Nanion people for their great help in our work. And finally, I would like to thank you all for your attention, and I am ready to answer your questions. And I can stop sharing now.
Thank you, Elena. That was a fantastic presentation. It's really exciting for me to see some of the polished data that's coming out of your research. That's wonderful. Let's see. Yaimar, are there any questions in the chat yet? I don't see.
Yes, we have one from Marcus. So he just raised the hand.
Okay.
I think you can formulate it. Yeah, exactly.
Hello everybody. I'm Marcus from Nanion Technologies. Thank you, Elena, for this very fantastic talk. I have a question related to the insertion of the Channelrhodopsin for all of them, actually, because you put in the protein in a eukaryotic cell system. Is there anything known interaction of cell metabolism and the Channelrhodopsin that could modulate the activity, like phosphorylation or lipid metabolism? Or
yeah, that's a very interesting question, but unfortunately not well investigated yet, because what is known that Channelrhodopsins in native cells, in algae or whatever protists, they are phosphorylated there. And this phosphorylation is reversible, and it is needed to regulate the adaptation, because just like our eyes, there is some adaptation to the light level, and it has been shown that this is achieved in the alga by reversible phosphorylation of Channelrhodopsins. But what happens in animal cells, I think nobody knows. And this is something that needs to be investigated. And definitely there must be some influence from the membrane lipids, because we know again from in vitro studies that Channelrhodopsins are very sensitive to the lipid composition of the membrane. In particular, their photocycle, and this means photocurrent kinetics, is altered. It depends on the lipids, so this has to be considered, but you don't have much freedom with animal cells, right? So they have some specific lipid composition, and you have to work with that. You cannot vary it at your will. But yes, all these factors are really important. What is even more important, that we know that in the alga, there are very likely there are other ion channels, calcium ion channels, not yet identified, that are activated by Channelrhodopsins. And that really increases the amplitude of currents that are involved in phototaxis. But those channels are not yet known, and that's a great possibility for future research. So if those channels can be found and transferred to animal cells, there is a possibility to greatly increase the efficiency of channelrhodopsins in animals. Yeah.
Thank you so much, Elena, for the answer. But so you mean like they could potentially form like functional, you know, that is sometimes known that calcium channels would interact with the calcium activated potassium channels, so they form like a how is it called? Yeah, like couples, you know, like a voltage gated channel that you have the membrane potential change very close to a voltage sensitive channel or something like that.
Yeah, about these putative channels, calcium channels, we don't know much, but we know that they are not voltage gated.
Ah, okay.
But how exactly they are gated, we don't know. Due to photoactivation of channelrhodopsins somehow leads to their activation. It could be direct protein-protein interaction, for instance, or it could be a pH decrease that channelrhodopsins would induce, something like that. But we don't know, and it's very difficult because the only information we have about them is from just physiological studies on the entire algal cell. And this is very difficult to interpret the results because, yeah, there are many things and they all interact. So it's hard to disentangle this. So we need these channels to be cloned, but so far all attempts have been unsuccessful.
Yeah. Because
there's proteomics done on these cells, nothing. Of course, the genome is fully sequenced of the model LG. It's again, well, there are many calcium channels, but nothing looks like good candidates for this role.
Interesting. Thank you, Elena. I think we have another question that came in from the chat, and it has to do with analysis. It says, how do you perform data analysis of SyncroPatch data?
Okay, that's a very good question, of course. And Nanion provides a software for analysis of this specific format in which recorded data are acquired. And this, well, at first, because we have this low rate of expression, so there were not really so many cells. So at first, I just exported Nanion data to patch format and analyzed it with this pclamp software that I used to use for manual patch clamp. But now I'm trying to learn how to use Nanion's analysis software, and they also keep developing it. So in principle, for standard experiments, you can just use Nanion software, which is called data control. And it would be, I'm sure it would be sufficient because you can calculate current voltage relationships automatically with them. And there are also options for this current clamp, like action potential generation and its parameters and things like that. Our research is different because it's quite non-standard. What we are doing is not what was originally intended by Nanion, because they designed their instrument for drug research, not for channelrhodopsin research, obviously. So it's a little bit something that we need to develop, but we are developing, and they also are developing, helping us with this. Yes.
Okay, thank you. We have one more question just came in, and it's about your cell prep. They say it's about the 10-centimeter dish. It says, you mentioned that you can use one 10-centimeter dish for these experiments. They are asking, how many cells do you get from this dish, and what enzyme do you use for the digestion?
Okay. Again, Nanion provides a detailed cell dissociation protocol, and they strongly recommend using the enzyme called EEE, which is a trypsin analog from some fungus, I believe. And that is the one because of course preparation of the cells is a key to success in this measurement. So it's really important that all cells are individual. They do not form any clumps, because clumps are useless for this technology. And well, I don't remember how many cells are there, what are the cell densities, but I just, it's something like maybe 80% confluent cell dish is sufficient. Tim, you probably better remember what are the cell densities in this case, because
the cell densities that are recommended depend on the number of holes. And so we recommend a minimum cell density of 100,000 cells per mil for a single hole, and it scales directly for 4 and 8 hole. So we would ask you to shoot for 400,000 cells per mil for a 4-hole chip, for example.
Yeah.
But if you are prepping cells from a 10 centimeter dish and you're getting the success rates that you're showing, that indicates to me that you have achieved that threshold.
We did calculate it in our first experiments, but then we don't do it every time because it's more or less consistent. I mean, if you see that they are 80% confluent, you know how many you get.
Yeah, exactly.
But it all, of course, depends on the cell type, because they are all different. They can be dissociated to a better or worse level and so on. So all this requires a little bit of trial and error, as usual, as it usually is.